Hello, everyone, and welcome to this week's edition of the Extracellular Vesicle Club. This is an event of the International Society for Extracellular Vesicles, and my name is Kenneth Whitwer. I was newly nominated and confirmed as the president-elect of the society, and I'm joined many weeks by Clotilde Thierry, our past president, and Metka Lenasi, who is our new chair of science and meetings for ISEP. But we do this event every week to try to highlight some of the recent publications, preprints in the field that many of you want to learn more about and talk about. So this week, we have a presentation coming to us from Vanderbilt University. And so Banishika Barman is going to be presenting her recently published work. And this was done in the group of Alyssa Weaver. And so Alyssa is also here with us today. So I'd like to welcome both of you. Alyssa, would you like to introduce Banishika, please?
Yeah, that would be great. So, Banishika, who also known as Bibi, so I'm going to call her Bibi because it's easier. She joined my laboratory in 2016, and as a postdoctoral fellow. Previous to that, she did a PhD in biochemistry at the University of Calcutta, really in one of the prestigious scientific institutes there. Under the direction of Dr. Suvendra Bhattacharya. And she did some very interesting work there on how the RNA silencing machinery is assembled, and showed that it's assembled, on mRNA associated with ribosomes at the endoplasmic reticulum. And she went on. That was a first author paper, and she went on in a couple of co-first author papers to show that trafficking and alterations in interactions with different organelles was involved in both degradation of the mRNA and recycling of pieces of a RISC complex. And so really, when she came to my laboratory, she was very interested in this idea that the processes that she'd been studying also involved transport of RNAs into extracellular vesicles, this juxtaposition of endoplasmic reticulum with other organelles and assembly and disassembly of RNA complexes. And so that's what she really did. And she's done a tremendous work that she's going to talk about today. I will just mention she's was recently promoted to instructor in my laboratory, but she will be looking for tenure track faculty positions very soon.
Thank you, Alyssa, for this kind of introduction. So now I will be talking about my recent work. Thanks, Alyssa, again for this kind introduction of me. So I'll be talking about VAP-A and its binding partner, CERT, drive biogenesis of RNA-containing EVs at ER membrane contact sites. So this was published in April 25th in Developmental Cell, and I will be talking about basically this publication. So as you know, that extracellular RNA has come out from many routes. So it can be found in large EVs or small EVs or with apoptotic bodies, or as just free ribonuclear protein, or exomeres or LDL with LDL or HDL. So the extracellular RNA comes in various forms. Today I will be talking about only this large EV bound and small EV bound small RNAs. So as you know, that RNA incorporation into the extracellular vesicles is very poorly understood mechanism. What we know from the previous reports that it could be a SCART dependent, where different SCART proteins come into play with the accessory proteins, and they help to shorting of the ubiquitinated proteins into the ILPs. Another route is tetraspanines like CD63, CD81, and CD9 dependent formation of ILPs, and where that different lipid enzymes is playing a role in this process. And third one is sumo-related HNRNP A to B1 related RNA sorting. So where in this report, they showed that the SUVO-related HNRNP-A2B1 basically recognize exomotif, a specific motif in the microRNAs, and that helps to get into those microRNAs to the MVPs and form ILPs and then secreted into the exosomes. So we wanted to look this that where are these mechanisms, where are that mechanism are. From our group, there was one paper that showed that Argonaute-2, which is a risk core component, it found in the exosomes, and that AGO2 interactions with the endosomes and secretion into exosome depends on the KRAS signaling. Also, there are one review that showed that the nucleation of AGO2 risk occurs on the rough endoplasmic reticulum. They also found microRNAs on rough endoplasmic reticulum, and that multivesicular bodies are probable site for the risk recycling. In another report, we found that the membraneless RNA granules, which is formed by TIS11B RNA binding protein, that meswork intertwined with the ER, and that particular domain on the ER is called tiger domain. So these are the studies that let us think that maybe there are some role for these endoplasmic reticulum to contact with the multivesicular bodies and to form exosomes or extracellular vesicles. So when we search different literature, we found membrane contact sites, that is the contacts between two organelles, and they are in very close proximity. They basically exchange repeats, and interactions between proteins can control vesicle fusion or organelle fusions. This contact between two different organelles could allow transfer of distinct RNA or RNA binding protein complexes to extracellular vesicles. So our hints was P-bodies, stress granules, endoplasmic reticulum, or nucleus, because these P-body proteins or stress granule proteins or ER proteins are found in the extracellular vesicles. But we first look into the endoplasmic reticulum. So to find out that ER and in MPP contacts, whether there is any contacts in between ER and APP or not, so we take a advantage of proximity ligation assay. So in this case, the GFP661 beta, which is the ER marker, we overexpress that GFP661 beta in DKS8 colon cancer cells, and we chose two primary antibodies, one for ER, that is KDEL, and another for MPP marker, that is CD63. If these two antibodies are close enough, that is up to 40 nanometer, they will generate a signal after reactions, and these signals come looks like a red dots. In this picture, you can find that red dots. These are the close approximation of these two markers, like ER marker and MPP marker, and the green one is GFP61 61 beta marked endoplasmic reticulum. So we can say from this one that ER and endosomes are in close proximation. So similarly, we wanted to look that whether microRNAs are also in these membrane contact sites or not. So for that one, we also did a proximity ligation assay in a different way, where we overexpressed MFP488B100 or MFP488 LET7A overexpressed in DKS8 colon cancer cells, and we choose the same antibody for ER and MPP like KDEL and CD63, and you can find if they are in close proximation, it will generate these red dots, and these green dots are either B100 or LET7A. And from this line of scan, you can find these are overlapping to each other. That means that these microRNAs are on the ER MPP membrane contact sites. So we also did electron microscopy tomogram for ER MPP contact sites. You can see from here, this is the MPP, and these are ILPs, the blue one, and this purple one, the endoplasmic reticulum tubule. We also did a video for that one. You can clearly see that these ILVs are touching to the ER and MPP membrane contact sites. However, we move. So now we are looking for the key protein that is responsible for these ER MPP membrane contact sites. So we found that VAP-A, or vesicle associated membrane protein associated protein A, is a key protein in ER endosome, ER membrane contact sites. So this protein can found in different contacts, including ER endosome MCS, but it also contact with ER PM or plasma membrane, ER lipid droplet, ER Golgi or Golgi, ER mitochondria. So we took this protein for our study, and we also found that this VAP protein interact different endosomal protein, which are OSBP, STAR-D3, ORP1L, all are related to the lipid transport. So next we knock down that VAP-A indicates 8 colon cancer cells and check the ER membrane contact site formation is okay or it is hampered. So we count that PLA dot in scramble control and VAP-A knockdown 1 and knockdown 2. We generally used 2 knockdown from 2 shRNA. And we count that fluorescent dots per cell, and we found that when we knock down VAP-A, the ER MPP contact sites formations are hampered significantly. We also observed that one by the TAME analysis. So this is a TAME picture for control, and this is from the VAP-A knockdown. And we measured that ER to MPP distance by nanometer, and also measured that percentage of MPP with ER contacts. And we found in VAP-A knockdown cells, both are reduced. The percentage of MPP with ER contacts are reduced, and the distance in between ER to MPP is increased because the contacts is lost due to VAP-A knockdown. So next, we try to find out whether this is hampering or manipulating that EV biogenesis or not. So we first check vesicles number per cell per hour by nanoparticle tracking analysis in small EVs and large EVs isolated from VAP-A knockdown 1 and knockdown 2. And we found that number of vesicles is significantly reduced. We also count the size of small EVs from our TAME analysis, and we found that the VAP-A knockdown cells has smaller vesicles in VAP-A knockdown cells. We also quantified total RNA per EV, and we found both in case of small EV and large EV, the total RNA per EV is reduced. So from this, we understand that there is something is going on due to VAP-A knockdown, that EV biogenesis is hampered. So next, we did a small RNA sequencing, and from A and B, you can see that PCA for microRNAs in small EVs and PCA for microRNAs in large EVs. And you can clearly see that scrambled and the knockdown 1 and knockdown 2 is well separated in both the EVs. That means there are differences in between microRNAs in VAP-A knockdown EVs in compared to the control EVs. So we did a normalizations with the cell EVs. So if there is any difference due to VAP-A knockdown in cells, so we normalize that to the EVs, and we found that there are a couple of microRNAs that are up or down regulated in the small and large EVs, and this heat map is showing the common microRNAs that are both down or up in small and large EVs. You can see from here, these are the common microRNAs that found in both EVs that are affected by the VAP-A knockdown. So we further tested some of these microRNAs by the qRT-PCR from this list, and also from because this list is we took that we made that heat map for that log 2-fold changes. So there are some other microRNAs also, those are not log 2-fold changes. So we know those microRNAs. So we checked some of those which have previous report to sorted differentially in DKS8 colon cancer cells. So we took some of the microRNAs from that list, from this list, and we found that these microRNAs are down in VAP-A knockdown small EVs and large EVs. Whether in cell, either they are non-significant, they are not changed, or some of the microRNAs are increased. So that means that that VAP-A knockdown is doing something for the microRNA sorting to the VAP-A knockdown extracellular vesicles. So we tried to find out that the subpopulation of the small EVs that is actually responsible for these changes, because we found small changes but significant changes in total RNA quantity and the small EV numbers. So we took a previously reported method in TNS in 2016. So we took that method for light and dense small EV purification. This is basically a high-speed purification for 60 minutes, that is 350,000 G. And this F3 fraction is the lighter fractions, and these F5 fractions is the dense fraction. So we run with this EV purification method, and we probe for some of our markers along with some of the RNA binding protein. And we found that these fifth fractions and the third fractions has differentially segregated proteins like AGO2, flotilin 1, HNRP, A2B1, LC3, VAP-A. These are mostly enriched in the denser EVs, whereas CD63, TSG101, syntanin, and ALIX are mostly enriched in the lighter fractions. This was also shown in another cell line, which is H1080 cell line, and we found the similar results. So when we measure the small EVs per cell per hour in light and dense EVs, we found the dense EVs always have smaller quantity of EVs compared to the light EVs, but very surprisingly, that dense EVs has more total RNA in compared to the light small EVs. That means we speculate these dense EVs actually possessing most of the RNAs. To check that, we also did TAME analysis for the scrambled and knockdown light and dense EVs and their size. So we found that the knockdown dense EV is smaller EVs than the control dense EVs, but there are not such difference in between light and denser EVs. Next, we did QRT-PCR for these scrambled and knockdown light and dense EVs, and we found most of the changes for the microRNAs are happening inside dense EVs. So we found that knockdown dense EVs, all of the microRNAs are going down in compared to scrambled dense EVs. But in case of light small EVs, there are no such significant change. We also checked whether these EVs, like dense EVs and light EVs, are actually full like lipid enclosed EVs or not, or RNA inside that EVs or not. And we did a RNA treatment plus minus detergent, and we found all the microRNAs are inside this either light EVs or dense EVs. So they are not sticking to the outer membrane of the EVs. So next we tried to find out that functional assay by the 2D co-culture, or just adding the small EVs directly to the cell, donor cells. So we took donor cells, that is the DK1 cells or small EVs isolated from the DK1 cells, And recipient cells are decayase 8 cells, which is expressing either luciferase control or luciferase mid-100 binding site. So we found that when we add that scrambled, so we have a couple of like donor cells. One is no donor, one is the same donor as the recipient cells, that is decayase 8 donor, and this scrambled or scrambled plus anti-mid-100 and scrambled plus anti-mid-100. So we can find these changes here to show that these cells are donor cells are actually releasing mid-100 to using this anti-mid-100. In C panel, you can see that AC donor cells, which is decay over donor cells, when is adding, so the luciferase is going down, but when we are adding knockdown 1 or knockdown 2 donor, is going up. So these two donor cells have less amount mid-100. We also checked the relative mid-100 levels in these EVs, and we found, yes, there's knockdown 1 and knockdown 2 DK1 cells release less amount of mid-100. We then tried with adding back the small EVs isolated from these cells. There's knockdown cells or the control cells. We found a similar result. We also isolate light small EVs and dense small EVs from these donor cells, and we found that light small EVs have no effect on these mid-100 reporter, but dense small EVs has actually the same effect that we found in the C or found in the E. So that means that dense small EV is possessing that mid-100, and this is the site where that all VAP-A knockdown effect is going on. So we then take some imaging tool to check that whether actually RNA binding proteins and RNAs are inside the MPPs or not. So we took GFP RAP5Q79L, which is a mutant versions of RAP5. So when you express that RAP5Q79L, you can find a larger vacuous kind of MPP. So it mimics the MPP structures. Otherwise, it's very difficult to find a MPP with the imaging system. And we overexpress that GFP RAP5Q79L and stained with CD63 for both scrambled control and the knockdown one. And we found that the size of the RAP5Q79L is down for the knockdown, but the number is high. That means they are not matured enough to mature enough to hold that CD63 inside them. And also we measure the percentage of CD63 inside that RAP5Q79L marked MPPs, and we found that level of CD63 is going down with the VAP-A knockdown. So that means the formation of ILBs is hampered by VAP-A knockdown. We also check 2 microRNA Cy3 mid-100. I am showing only Cy3 mid-100, and also AGO2 and SYNTIP. I am showing only AGO2. So you can find this is scrambled one, and you can find this red Cy3 level mid-100 inside the scrambled one. But when you can see in the knockdown, these are not inside that. The red dots that is mid-100 is not inside that MPP. Similarly, you can see that these red dots, which is AGO2, inside that GFP RAP5Q79L along with CD63. But when you can find these knockdown, there are no such AGO2 inside the MPP. So we also quantified that here, and we found the significant changes of AGO2 and Cy3 mid-100 in that percentage of percentage of Cy3 mid-100 or AGO2 inside RAP5Q79L, and in right side, this is the intensity of that mid-100 or AGO2. Both are going down with the VAP-A knockdown. So next, we found a literature where this showed that ceramide is responsible for negative curvature of endosomal membrane, and these negative curvature actually form that extracellular vesicles. So we found it very interesting, and we tried to check that whether the ceramide level is okay with that VAP-A knockdown or not, because VAP-A bind to the different lipid binding proteins. So we did untargeted lipid mass spec for VAP-A knockdown small and large EVs. So we found there are multiple sphingolipids, including ceramides and sphingomyelins, that is actually going down for the VAP-A knockdown EVs. We also checked with the targeted lipid mass spec with the C16, C22, and C24.1 ceramide molecules, and we found all these molecules as going down in the small EVs or large EVs, but in case of cell, these ceramide molecules are unchanged. So that means that the formation of EV by ceramide is also affected by the VAP-A knockdown. So that CERT or ceramide transporter protein, which actually interact with the VAP-A, they say whether the VAP-A regulates ceramide transporter protein that trafficking to the extracellular vesicles. So this is the structure of CERT molecule, and it interacts with the VAP-A. So we stained we overexpressed that GFP RAF5-79L again and stained with CERT and CD63. So we found that CERT and CD63 are both inside that MVB, but when we knocked down VAP-A, it hampered the CERT localization to the MVB. This is also found in this quantification, that the percentage of CERT inside RAF5-79L is going down, and also the intensity of CERT is going down. So next, we did we overexpressed GFP VAP-A and mCherry RAF5-79L, and also stained with NSMS2, because you know that NSMS2 or neutral sphingomyelinase-2 is required for the ceramide formation from sphingomyelin. So we stained all these three, and we found that. So this is the gray is the MVB, red is the NSMS2, and green is the GFP VAP-A. So now it's converting to 3D, and you can clearly see that GFP VAP-A and NSMS2 is very close to each other, and they are touch, and NSMS2 actually not inside that RAF5-79L, they are just out touching the limiting membrane of the MVB. Yes, here. So our model is that VAP-A on endoplasmic reticular membrane interact with the CERT, and these interactions brings ceramide either from the endoplasmic reticulum where de novo ceramide synthesis occurs, or somewhere else where sphingomyelin is converted to the ceramide by NSMS2, and they form that membrane curvature or negative membrane curvature to form that ILP, and to form that ILP, and in the same time that RNA binding complex which are close to the ER membrane, or RNA granules which is also close to the ER membrane. From previous study we know that they are bringing that RNA and RNA binding complex to this MVB and form that ILP to secrete in the secrete to the outer space as a extracellular vesicles. So this is our model for that one. So for our study. So I thanks to my mentor, Professor Alisa Weaver, to Bong Hwan, especially because he helped for this imaging a lot, and all other collaborators who are part of this project, and also other members from our lab and our fundings and our department. So if you have any questions, please go forward.
All right. Well, thank you very much, BB, for this presentation and for this very interesting work, which I think is quite relevant for all the questions about how does RNA get into EVs and what happens with it. So we do have some questions in the chat box that have come in already. So I'm going to get to those, and I'm going to allow unmuting now. So when I call your name, please go ahead and ask your question. So we have several questions actually from Stacia Nicholson. So Stacia, could you please go ahead and ask your questions? I think some of them you were asking about specific slides, so feel free to just remind us of which were which, because we just don't have a mechanism of doing it in real time. So we just wait until the end of the call.
Yes, no problem. Thank you. Yeah, I was trying to see if there was a slide number, but I don't know if I missed it or not. So I was just trying to reference which one it was. But the first question was about the, you showed, I think it was a TEM or some type of, I don't know if it was actually TM, but it was an image of the multivesicular vesicle. And I was trying to see if it was captured with, if it was that was captured within the cell, or if that was captured somehow external to the cell, where it was located. Because I know when literatures that I was reading, they were talking about people haven't been able to capture MVBs outside of the cell. So I was interested to see if you were able to do that.
You are talking about
that guy.
This one?
Yes.
No, this is the whole cell. So
this is the whole cell. Okay.
Yes. So this is inside the cell.
Okay. Yeah, I was just wondering because I wasn't seeing at first any other.
I think she may be confused because of the way the tomogram is colorized and then extracted.
Yes.
Okay. My second question was, how did you sort the small and large EVs? How did you separate them? I think I might have missed that.
So generally we do Cushon density gradient. So that is 100,000 G over the cushion, and then take that cushion and do that density gradient for the overnight. So after overnight washing, and then we get the pellet of small EVs, and large EVs is just 10,000 G pellet.
Okay, so it was with the density gradient centrifugation, because I was just wondering if maybe you used SCC, or would you have any suggestions about if that's possible to use SCC for that, or is the density gradient the best method for that?
So for our cells, we do generally density gradient that is more purified, and it's laborious, but it's more purified EVs we can always get, and we check with the EM that the structure of the EVs is fine. So we prefer density gradient. Also for the RNA, because you know that there are also RNA particles coming out outside EV, so prefer that cushion density gradient. But if you're the initial sample is too low to do a density gradient, then you can do SCC.
Okay, because I work with biofluids, and we have very small sample volumes.
Case,
in
that case, it might be SCC is the best for you, not for not density gradient. It's difficult to do density gradient for biofluids.
Great. And Stacia, did you have one other question?
RNAs.
Yeah, I think you may have just cut out there. So if you just want to start over again.
Okay, yeah, I was actually going to ask the one last question. Thank you. It was about the slide with, I believe it was the RNA protection assay, to show that
Yes.
the microRNAs were from the EVs. Can we see that slide again? Because or can you just like go through your method for that? Because I completely was overthinking while that was going on, and I think I missed some important information. Because I was working on an RNase protection assay for my lab, and I was sending samples to the bioanalyzer for quantitation, but then I was never getting complete knockdown with combination treatment with detergent and proteinase K. So then I saw that some people were using qPCR to analyze RNAs that were specific to extracellular content as well as to ones that were specific to EV content. So yeah, I was just interested in how you did that.
Okay. So this one. So we didn't use that proteinase K or any proteinase. So we did RNase treatment with the plus minus detergent, like we use Tritron X100 for the detergent, and we incubate with that detergent and RNase, or as a control without detergent plus RNase, or plus minus RNase minus detergent. And this is a 30 minutes incubation in 37 degree. And then you can do like enzyme inactivations and isolate RNA and do qPCR. Yes.
Okay, so the qPCR is the best bet, because that's what I was thinking, because with the other one, like we were seeing like lower levels in the different comparison groups, but there was never like any complete knockdown. But I did see differences in the microRNA, the small RNA percentages that gave me the indication that we were having differences in internal EV microRNA loss versus what might have been exogenous. So I was trying to see if that was true. But, and yes, I found that paper, and I was just recently working on like the qPCR process. So I'm glad to see that that's what people are doing. So I'll continue with that. Thank you very much. That's it for me.
Thank you. Thank you for the questions. All good questions. And if I could just follow up on that, what about protease? I mean, do you ever do a protease plus?
We didn't do protease yet, but we are thinking to use protease as well in combination with RNAs, but we didn't do it yet, so we don't know the answer.
Very good. Very good. Well, you've thought of everything. Let's see. So who else do we have here? Moritz Weigel.
Thank you. So thank you for the great presentation. I was just wondering regarding the co-culture experiment that you showed. Did you also, you showed very nicely that the luciferase reporter is responsive in the recipient cells to the transfer or to the VAP-A knockdown. And I was just wondering, did you also quantify microRNA 100 levels in the recipient cells, or did you also try to quantify in terms of the copy numbers the number of molecules that are being released, and how that compares to the number of microRNAs that you find in these recipient cells?
Okay, so we did quantify mid-100 in the recipient cells, but we didn't do the copy number for recipient cells or donor cells. So we don't know the answer of copy numbers, but yes, we did check by qPCR the level of 800 in control and VAP-A knockdowns.
Okay, thank you. So, and you did see the increase in the recipient cells?
Yes, yes.
Yeah.
All right. Look at the host being not so adept with the controls here, but thank you, Moritz. We now have a question from Anand Sasidaran. Hi, Anand.
Hey, hi. Hi.
Go ahead.
I lost my mic. I hope you guys can hear me.
We can.
Really a great work. I have a quick question. Have you actually looked into, so there's been a paper talking about the exomotive microRNA, which is very specific for vesicles, and the cell motif microRNA, which is very specific for cells. So have you found these microRNAs in your study? And have you seen a reduction of exomotif microRNAs when you knock down VAP-A?
So we didn't do that like exomotif or the cell motif comparison, but yes, some of the microRNAs that that was mentioned in that paper. So some of the microRNAs is also in my like data set.
So
but we didn't do that like analysis by ourselves, but I just took that like their microRNAs which has exomotif and altered sorting for the cell for the EVs. So it matched some of the microRNAs in my data set after
So
sequencing.
just to clarify, which exomotif paper are we talking about here? Because there are
I
a
think
few.
he's talking about that
The Ronald Khan's paper, Ruben, Grisha, Martin's first author.
Okay, so yeah, I think the two of you are talking about different papers.
Okay, sorry.
So I think that,
checked the Villaro-Peltri's paper for that, which one, NatCom paper? So exomotif, that RNPA2B1 paper.
Yeah, so you were looking at the 2012 paper versus the 2021 paper.
21. I asked 2021.
Okay. Sorry.
Great. Yeah. So that maybe that's something worth looking into as well, whether any of those motifs are found. So yeah, that's a very interesting question. And I want to, I don't think that we have any other questions here. Anand, did you follow up on that? Yep, go ahead.
So you did knockdowns of certain genes and then looked for your vascular small RNAs, right? Do you actually find, when I say small RNA, I'm talking about the whole population of small RNA, not just microRNAs. Do you find an upregulation of small RNAs when you do a knockdown and you look inside EVs? Not just microRNA, the whole RNA. So
can you repeat your question, because it's your sound is breaking.
Oh, I'm sorry. So 1 second. Okay. When you do a small RNA, when you do a gene knockdown in your system, I assume you're doing in cells, do you actually find an upregulation of small RNA population inside EVs?
Small RNA population in EVs?
Yeah, not microRNA as a whole.
Oh, okay. So we did small RNA sequencing, and we do have data in my supplementary figures if we want to look at. So we have SNO RNA data and the tRNA fragments data that are altered for the VAP-A knockdown. So this is not only microRNA, it also like SNO RNAs and tRNAs that are affected by the VAP-A knockdown in small EVs and large EVs. So we do have that data. You can check in my supplementary figures, yes.
And have you ever thought of doing an organoid pull down from your small EVs to see what are the associated microRNAs and complexes inside it?
Sorry?
Have you ever planned to do an organoid pull down from your EVs just to understand what are the associated active microRNAs suppressing your construct inside the vesicles? Have you thought about it?
I mean, yes, we can do that, like to pull down RNA from the small EVs, but I mean, yes, that is in our list, but as like quite like challenging, I think, pulling down RNA from the small EVs or large EVs, yes.
Thank you, amazing, thank you.
Yeah, that would take a lot of EVs.
That would take, yeah, that's a lot of material, for sure.
Yes.
Well, good. So yeah, thanks everybody for the questions. I think we've reached the end of what's in the chat box right now, but I want to just follow up with something. In fact, I was just chatting with somebody about this by email. So you've identified this population of relatively RNA-rich EVs. Do you envision that there will be some way of obtaining this population easily in bulk, either from cell culture or from a biofluid? Do you think that's possible for us to obtain, through whatever method, a large enough amount of this stuff that we could check its function?
Okay, so that is what we are right now looking for, that how we increase that level of density, because it's very low. So we are trying to find out a way by 3D culture or something that to make that level of density with higher amount, so that we can do more study on that. Because
good. Yeah, that's great. I mean, I see that Phil Askinase has asked the same question. I mean, a similar question. Are there distinct markers on the dense EVs that you could potentially use for this?
Yes.
Yeah.
And what are they? What are the markers on the dense EVs? So
for the dense EVs, I mean, there are no such markers yet, but we found that the flotilene is enriched in dense EVs, but we didn't do any proteomics for these dense versus light EVs. So we don't know yet that what are the markers for these dense EVs, but we are working on that to find more and more about these dense EVs in terms of proteomics and in terms of RNA.
Very cool. All right. Well, well, I'd like to thank BB, you for presenting today, and Alyssa for also, I guess, initiating this work and supervising, and for also your remarks on the center that you've established so well at Vanderbilt. So thanks everybody too for joining EV Club today, and we'll look forward to seeing you at an event coming up very soon. So take care, everyone. Good luck with your research, and
Thank you.
talk to you soon.
Thanks, Ken.
Bye now.